Hello all. Today we will talk briefly in some enzymes that use in cloning technology. First of all, the famous enzymes that use in cloning is the restriction in the nuclease. What is restriction in the nuclease? It's an enzyme produced by bacteria as innate immunity from the bacteria against the invader. There are three types of restriction in the nucleases that make it useful in the cloning. The first is isochiazomers, such as VSPEI, which is produced from Bacillus species, and ACC3 from Actinobacter calcoacticus. The recognition type for these two examples of restriction in nuclease is only one. The product is the same. The other one is the new chiazomers, such as NAR-A or NAR1 from Nocardia argentinensis and SFO1 from Serita pantocola. Bind the same DNA sequence but cut at different sites, like here. This is the same origin or same loci, but the product is different. One of them is cutting sticky ends, and the other is SF1 is cutting like plant. The third one is isocodimers, such as NCO1 from Nocardia corallina and page 1 from Pseudomonas alcigenus. Bind different DNA sequence but produce the same sticky end. They are binding to the different loci in the target but produce the same sticky end or same product, like here, CATG. Other enzymes used in the cloning technology is the nucleases that cleave many of enzymes. Many other enzymes are used to prepare DNA for cloning in addition to restriction in the nucleases that degrade single-stranded extension, such as S1 nuclease and monk bean nuclease, are used to generate blunt end of cloning. Here, blunt end. This is useful when the recognition sequence of restriction enzyme that produce complementary sticky ends are not available on both the vector and target DNA molecule. Blunt end can also be produced by extending three recessed ends using a DNA polymerase, such as clean-out. Here, clean-out polymerase derived from E. coli DNA polymerase type 1. Another type of enzyme that called phosphatases, such as calf intestinal alkaline phosphatases, cleave the 5-phosphate group from restriction enzyme digested DNA, and a 5-phosphate group is required for formation of a phosphodiester bond between nucleotides, and therefore its removal prevents recircularization, self-ligation of vector DNA. Another one of enzymes is kinases. Kinases added phosphate group to the ends of the DNA molecule. Among other activities, T4 polynucleotide kinases catalyze the transfer of the terminal terminal lambda phosphate from the nucleoside triphosphate to the 5-hydroxyl group in the 5' end of polynucleotide. The enzyme is employed to prepare chemically synthesized DNA for cloning, such such DNAs are often missing a 5-phosphate group required for ligation to vector DNA.